Have you ever wondered why fluorescence markers are so crucial in real-time PCR? Well, these markers are like the shining beacons guiding the process of polymerase chain reaction, or PCR, a technique widely used in molecular biology. They play a pivotal role in identifying and quantifying DNA sequences in real time, making them indispensable in both diagnostics and research. These markers work by emitting light upon binding with the DNA, providing a glow that signifies the presence and the amount of the specific DNA sequence being investigated. This real-time tracking of DNA amplification is what makes real-time PCR a powerful tool in the world of genetics and microbiology. From identifying viral infections to genetic mutations, and from diagnosing diseases to research in gene expression, fluorescence markers are the unsung heroes, silently illuminating the path for every breakthrough. Now that we understand their importance, let's delve into the different types of fluorescence markers used in real-time PCR. Opening. First on our list is the TaqMan probe. Delving into the specifics, the TaqMan probe is a type of hydrolysis probe. It carries a reporter dye, usually fluorescein, at its 5' end and a quencher, tetramethylrhodamine, at the 3' end of the oligonucleotide. Under normal circumstances, the probe remains coiled, bringing the fluorescence dye near the quencher and inhibiting the fluorescent signal. The oligonucleotide of the Taq polymerase has a region homologous with the target gene. So when the target sequence is present, it binds with the sample DNA. As the Taq polymerase begins synthesizing new DNA strands in the extension stage, it degrades the probe through 5' end nucleus activity. This separation of fluorescein from the quencher generates a fluorescent signal. As the process continues, the number of signal molecules multiplies with each cycle, increasing the fluorescence. This increase is directly proportional to the amplification of the target, making it a vital tool in the PCR process. But it's not all roses. One downside of the TaqMan probe is its inability to provide information about each amplification cycle or the melting temperature. Nevertheless, its specificity makes it a popular choice in the world of real-time PCR. Closing. That's a wrap on TaqMan probes. Now, let's move on to SYBR Green. Now let's turn our attention to SYBR Green. This intriguing fluorescence marker is a dye that emits a strong fluorescent signal when it binds non-specifically at the minor groove of DNA. It's like a beacon of light in the dark, illuminating the DNA sequences it binds to. SYBR Green is somewhat of a superstar in the world of fluorescence markers. Why, you ask? Well, it's all down to its high signal intensity. Other fluorescent dyes like ethidium bromide or acridine orange could be used, but SYBR Green outshines them with its stronger, more prominent signal. But it's not just about the strength of the signal. SYBR Green also has a unique advantage over the TaqMan probe. It can provide information about each cycle of amplification as well as about the melting temperature. This additional data is not obtained from the TaqMan probe, making SYBR Green a more comprehensive tool in certain situations. However, it's not all roses with SYBR Green. Its main drawback is its lack of specificity as compared to the TaqMan probe. While it binds to the DNA, it does so non-specifically, which can sometimes lead to less precise results. Despite this, SYBR Green is a powerful tool in real-time PCR, a tool that, when used wisely, can provide invaluable insights into the world of DNA. That was all about SYBR Green. Let's now summarize the key points. We've learned quite a bit about fluorescence markers in real-time PCR. Let's take a moment to summarize. TaqMan probes, hydrolysis probes with a reporter dye and a quencher, behave like shy performers under normal conditions. They coil in on themselves, keeping the fluorescent signal of the dye quiet. But when the target sequence is present in the mixture, it's showtime. The probe binds with the sample DNA. And as the Taq polymerase starts synthesizing new DNA strands, the probe degrades, separating the fluorescein from the quencher and allowing the fluorescent signal to shine through. As this process repeats, the fluorescence increases, indicating the successful amplification of the target. On the other hand, we have SYBR Green. a dye that emits a strong fluorescent signal when it binds to DNA's minor groove. It's preferred for its ability to provide information about each amplification cycle and the melting temperature, although it's not as specific as the TaqMan probe. That concludes our exploration of fluorescence markers in real-time PCR. Remember, these markers are the unsung heroes of our PCR process.